Last week we found a 47 kilodalton band in the Site 7 drainage samples that has no business being there. SDS PAGE tells me its size. It does not tell me its identity. If I want to know what protein I'm looking at, I need an antibody. Western blot is how I use one.
Western blotting, formerly immunoblotting, is a three-stage method. You transfer separated proteins from your SDS PAGE gel onto a solid membrane. You block the membrane against nonspecific antibody binding. Then you probe with antibodies to detect your protein of interest with molecular specificity. The antibody does what no gel stain can. It distinguishes one protein from thousands.
The gel tells you something's there. The Western tells you it's specifically yours. A validated primary antibody runs you $200 to $800 a tube. Welcome to the technique, where your reagents cost more than your equipment.
The principle depends on molecular recognition. Your primary antibody, typically a monoclonal or polyclonal IgG, binds a specific epitope on the target protein. Because your protein has been denatured by SDS, that epitope must be a linear sequence, not a folded three-dimensional structure. If your antibody was raised against a conformational epitope, it may not recognize the denatured form at all. This is one of the most common failure modes in Western blotting, and it rarely appears in the antibody datasheet in plain language.
It is mildly ironic that a technique designed to identify proteins works only on proteins that have been chemically destroyed.
Subscribe. Method Monday, every Monday. Last week we ran the gel. This week we find out what's actually in it. If the Site 7 band is what I think it is, the conversation next week gets considerably more interesting. Membrane choice. You have PVDF and nitrocellulose. Use PVDF. Higher protein binding capacity, up to 200 micrograms per square centimeter versus 8 to 100 for nitrocellulose. Chemically resistant. You can strip it and reprobe for a second target. Nitrocellulose tears if you look at it aggressively and cannot be dried and re-wetted. The cost difference is marginal. Use PVDF.
PVDF is hydrophobic. It requires methanol priming to render it compatible with aqueous transfer buffer, 30 seconds in 100% methanol, then immediate transfer into equilibration buffer. If the membrane dries after activation at any point, the surface chemistry is disrupted and protein binding becomes uneven. This is not a recoverable error post-transfer.
I activated mine earlier, before the SDS PAGE run finished.
Where is it now?
On the bench.
Is it in buffer?
No.
It's dry. Reactivate it. 30 seconds and 100% methanol. Straight into transfer buffer. Do not let go of it between steps.
PVDF surface binding depends on uniform hydrophobic interactions across the entire membrane. Partial drying collapses those surface regions. When protein reaches a dried zone during transfer, it either fails to bind or binds at aberrant density. The result is bands that are sharp in some areas and absent or diffuse in others, a pattern that looks like a transfer problem, but is actually a membrane preparation error. Reactivation is the only fix before transfer. There is no correction post-transfer.
It is mildly common that the membrane fails before the experiment begins.
Transfer buffer. Tobin. 25 mmHg TRIS 1 to 92. Memented glycine. 20% methanol. The methanol is doing two things. It strips residual SDS from the proteins to improve membrane binding, and it dehydrates the gel slightly to prevent its swelling during transfer. Do not exceed 20% methanol. Above that, the gel stiffens, and large proteins can't diffuse out. For proteins above 100 kilodaltons, some labs drop methanol to 10% and add 0.05% SDS back to the buffer to help large proteins elute. The 47 kilodalton Site 7 band is fine with standard Taubin.
The methanol concentration represents a compromise. Methanol promotes protein binding to the membrane by reducing SDS coating. SDS competes with membrane binding sites. But methanol also increases polyacrylamide rigidity and retards protein diffusion. The empirically validated window is 15 to 20% for proteins in the 10 to 150 kilodalton range. For very large proteins, the improved diffusion from lower methanol outweighs the slight reduction in membrane binding efficiency.
Tobin buffer costs approximately $2 per liter to make from stock. The alternative is pre-made transfer buffer at $15 to $25 per liter. The chemistry is identical. The choice is your time versus your budget.
Building the sandwich. This is where most people make a mistake they don't discover until the blot is blank. Orientation. In wet transfer, proteins carry a negative charge from the SDS. They migrate toward the positive anode. The membrane must sit between the gel and the anode. If you build it backwards, proteins transfer into the buffer tank. You get a pristine blank membrane and no indication anything went wrong for 2 hours.
Most commercial transfer cassettes use a color convention: black side toward cathode, red or clear side toward anode, gel on the black side, membrane between the gel and the red side. Confirm this for your specific cassette the first time you use it. The convention is consistent across most major manufacturers, but not universal.
Roll out air bubbles between every layer of the sandwich with a roller or a cold glass pipette. An air bubble is a gap where no transfer happens. You will see a white void in your band. Roll it out. Transfer conditions. Wet transfer at 100 volts for 1 hour with ice block or in a cold room to manage heat, or 30 volts overnight at 4 degrees. Overnight is more reliable for anything above 80 kilodaltons. Lower voltage, less heat, more time for large proteins to migrate. After transfer, before you do anything else, stain with Pinceau S. It's a reversible protein stain. You can see if your proteins transferred and approximately where they are before committing to blocking an antibody.
Pinceau S is underused. It stains all transferred proteins red within 2 minutes and washes off completely with water. It tells you immediately whether transfer occurred, whether it was uniform, and whether your protein of interest is in the expected molecular weight region. A failed or partial transfer discovered at the Pinceau step costs you 10 minutes. Discovered at the ECL step, after 18 hours of blocking an antibody incubation, it costs you the whole experiment.
Blocking. The membrane has protein binding capacity everywhere, not just where your target protein is. If you add antibody to an unblocked membrane, it sticks to everything. Blocking saturates those sites with a protein your antibody ignores. Standard, 5% non-fat milk in TBSD. That's tris buffered saline with 0.1% tween 20. 1 hour at room temperature with gentle rocking. This step is not negotiable in length.
The choice of blocking agent is not arbitrary. Milk contains casein, which is a highly phosphorylated protein. If you are using a phosphospecific antibody, one that detects a phosphorylated residue on your target, the casein in milk will compete with your phospho target for antibody binding and dramatically reduce signal. For phosphoantibodies, use 5% BSA in TBST. Bovine serum albumin is not phosphorylated and does not interfere.
I used milk for the phospho-AKT antibody.
How long has it been incubating?
1 hour. I'm ready to add the primary.
Don't. Strip it and reblock with 5% BSA. The casein has been soaking up your antibody binding sites. Any signal you get from this blot will be non-specific background.
Casein is phosphorylated at multiple serine residues. A phosphospecific antibody raised against, for example, phosphoserine 4-923 of AKT cannot distinguish casein's phosphoserine from your targets. The antibody is quenched in solution before it reaches the membrane. Signal to noise collapses to near 1.1. This is one of the few Western blot failure modes that produces not a blank blot, but a uniformly high background blot. The antibody lands everywhere, non-specifically, because the specific recognition is neutralized.
It is mildly consistent that the component of the protocol that looks least important is the one that determines whether the antibody can work at all.
Primary antibody. Dilute according to the datasheet, and treat that number as a starting point, not a final answer. Antibody lots vary. Your protein abundance may differ from the manufacturer's test conditions. If your signal is too high, dilute further. Too faint, concentrate up. Standard incubation, overnight at 4 degrees Celsius. On a rocker, you get better signal to noise than room temperature incubation, because non-specific binding kinetics are slower at 4 degrees. 2 hours at room temperature works if you're pressed. Overnight is worth the wait.
The primary antibody concentration is the single variable with the largest impact on signal to noise. At too high a concentration, both specific and nonspecific sites are occupied. High background obscures your band. At too low a concentration, signal falls below detection threshold. For a new antibody, run a dilution series, 1-500, 1-1000, 1-2000, 1-5000, before committing to a final experimental concentration. This takes one blot, but prevents months of unclear data from a non-optimized antibody.
A primary antibody tube used at optimal dilution and stored correctly lasts 50 to 100 experiments. Used at twice the concentration because someone didn't optimize, it lasts 25. At $400 a tube, suboptimal dilution is a budget problem disguised as a technical one.
Washes. After primary antibody, 3 washes, 10 minutes each, in TBST on a rocker. These remove unbound primary antibody. The 10 minutes is diffusion limited. It takes that long for unbound antibody to leave the membrane. Short washes leave antibody behind and produce background. After washing secondary antibody, your secondary must be raised against the host species of your primary antibody: rabbit primary, anti-rabbit secondary, mouse primary, anti-mouse secondary. Get this wrong, and you get exactly 0 signal.
Secondary antibodies are immunoglobulins raised against the Fc region of the primary antibody's host species. An anti-mouse secondary binds only mouse IgG. It cannot bind rabbit IgG. The error produces a membrane that looks technically perfect. No background, no signal, no indication that anything went wrong. You will not know until the detection step returns nothing.
My secondary is anti-mouse. The primary I just used was a rabbit antibody.
Do we have an anti-rabbit HRP secondary in the shared stock?
There's one from last year.
Check the expiry. Restart the secondary incubation with the correct one. Strip and rewash the membrane first.
The Fc regions of mouse and rabbit IgG are structurally distinct. An anti-mouse secondary has no binding affinity for rabbit IgG whatsoever. The failure mode is absolute, 0 signal, 0 background, a membrane that looks as if no antibody was ever added. This is why you confirm host species on both datasheets before you start, not after you get the result.
It is mildly systematic that the blank Western blot looks identical whether the protein is absent, the transfer failed, or only the secondary antibody was wrong.
Detection. Enhanced chemiluminescence. Mix the 2 ECL substrate components, 1.1, the luminol solution, and the oxidizing solution. Apply to the membrane and expose immediately. Start with a 1-minute exposure, too faint. Go to 5 minutes, saturating immediately. Try 30 seconds. You have about a 20-minute window before the signal decays. Work fast. If you're using X-ray film, have your cassette ready before you add substrate. If you have a chemiluminescence imager that's faster and more quantitative, use it.
ECL works by HRP-catalyzed oxidation of luminol. The oxidized luminol emits light at 428 nanometers. A chemical enhancer amplifies the reaction roughly 1,000-fold. Signal peaks at 5 to 10 minutes and decays over 20 to 30 minutes. It is proportional to HRP concentration, so proportional to secondary, proportional to primary, proportional to your target, as long as you stay in the linear detection range.
Standard ECL substrate costs around $50 for 100 milliliters. High-sensitivity substrates for low-abundance proteins run $200 to $400 for the same volume and extend your signal window to 60 minutes. Know your target expression level. If you're detecting beta-actin loading control, you do not need high-sensitivity substrate. If you're chasing a cytokine at femtomolar levels, you do.
Good Western. 1 sharp band at the expected molecular weight, clean background, ladder bands tight and evenly spaced on log scale. For a purified protein, 1 band. For a crude lysate, multiple bands. Your target should stand out by position or intensity. 2 bands where you expect 1. Check for known isoforms, post-translational modifications, or antibody cross-reactivity before declaring a result.
For quantitative work, include a loading control, beta-actin at 42 kilodaltons, alpha-tubulin at 55, GAPDH at 37, at a different molecular weight from your target so they co-detect on the same membrane. Unequal loading makes band intensity comparisons between conditions meaningless. This is not optional if you are publishing.
The The statistics for normalizing band intensity across conditions, variance, error propagation, what sample size you actually need before the number means anything, I cover that on Saturdays. Statistical Saturday. First episode this week.
You're running a statistics series.
Yes, I'm good at math.
That is genuinely surprising. I did not know that about you.
I did math once.
What happened?
I got high.
It is mildly predictable that Patar confused math with methamphetamine.
2 failure modes. High background across the whole membrane. Blocking was insufficient. Primary antibody concentration too high, or washes too short. Fix, fresh blocking buffer, more dilute antibody, longer washes. Ghost bands at unexpected molecular weights. Primary antibody cross-reacts with related proteins, or you have ECL bleed from an adjacent lane. Fix, titrate the antibody more carefully. Confirm specificity with a knockout control if available.
The logic of Western blotting is subtractive. You are not generating signal. You are eliminating everything that is not your target until what remains is specific. Blocking removes non-specific membrane binding capacity. Washing removes unbound antibody. Detection reveals what was left behind. Rush either elimination step, and what remains includes noise you did not remove. The experiment does not fail loudly. It fails quietly, with bands in places they should not be, at intensities that do not mean what you think they mean.
Right? The Site 7, 47 kilodalton band. I had access to a commercial antibody screening panel. 48 antibodies covering common environmental and industrial proteins, curated from a supplier I won't name at this time. I ran the Site 7 membrane against each one over 6 nights.
I ran all 6 overnight incubations. The 4th and 7th antibody in the panel gave signal. The rest were clean.
It is mildly significant that 6 nights of overnight incubations in a residential shed is a controlled experimental protocol by most definitions.
The 4th antibody in the panel is raised against a metabolic enzyme found in sulfate-reducing bacteria, specifically the dissimilatory sulfite reductase beta subunit. The 7th is against a stress-response chaperone upregulated under heavy metal exposure in gram-negative bacteria. Both are present in every Site 7 sample we have. Neither appears in any of the 4 control sites.
A sulfate-reducing organism expressing heavy metal stress response at consistent levels in an industrial drainage corridor for at least 6 months. That is either a bioremediation application worth filing on, or an environmental liability worth avoiding. Either way, whoever identifies the organism first is in a position to do something with that information.
Closing tip. If your Western blot bands are sharp but shifted slightly in molecular weight compared to your last run, check 2 things before assuming anything. Did your gel percentage change, and did your transfer buffer methanol concentration change? Both affect apparent molecular weight. A band that moves between experiments is not necessarily a different band, but it's also not necessarily the same one. Verify before drawing conclusions. That's Western blot. Transfer your proteins to a membrane, block non-specific sites, probe with antibodies, detect with ECL. The Site 7 sample is expressing sulfate-reducing bacterial proteins under heavy metal stress. Something biological is running in that drainage corridor, and it has been running for months. Next Monday, mass spectrometry sample prep. We cut that band out of a Kumasi gel, digest it with trypsin, and send it for sequence identification. We're going to know the exact protein.